Hello, everybody. My name is Florian Merkle, and I'm a principal investigator at the University of Cambridge at the Institute of Metabolic Science. Today, I'll be telling you about our group's work and some basic principles of studying obesity with human hypothalamic neurons. So I'm going to now share my screen and start the presentation. Great. So there we are. So before we start, I just wanted to highlight two key take-home messages that I hope you get out of this presentation. The first is that having access to human stem cell-derived hypothalamic neurons can yield novel functional insights into these cells, such as the production of neuropeptides and how these cells respond to hormones and other metabolic signals, and that each model system has its own strengths and weaknesses. There is no perfect system in the world, and if possible, the relative strengths and weaknesses should be used in a complementary manner to gain confidence in whatever is observed. So I think that we live in a very exciting time where there are these emerging resources and tools that kind of offer the possibility of dramatically increasing our understanding of how genes and environmental factors associated with obesity can lead to this disease. These include now this rapidly growing databases of human population genetics, the transgenic mice, libraries of drugs, but also the technologies such as CRISPR, both CRISPR knockout and CRISPR-I, CRISPR-A, single-cell RNA sequencing, which is revealing cell type-specific transcriptional signature and also responses to functional perturbations, high-throughput screens, and also tools that give direct physiological insights, such as genetically encoded calcium indicators, such as GCAMP, and electrophysiological methods. And traditionally, these have been used in animal models, which has been absolutely invaluable in defining the cell types and circuitry that regulates food intake and energy expenditure, and has defined also what are the behavioral and physiological responses to different diets and to different perturbations. And together, these have really led to some insights and treatments, but it has been slow going, and there just isn't a broadly effective kind of population level treatment for obesity and diabetes, although there are promising drugs in the pipeline, and I think we are definitely improving. So what our vision is that we can harness the strengths of these animal models, but also complement some of their weaknesses by using stem cell-derived hypothalamic neurons. So as I'll show in the next slide, these have several advantages. So they can deconstruct some of the complexity that exists in the brain, where there are many cell types and that are deeply interconnected with each other. They're also amenable to the performing of genetic and drug screens to generate kind of a higher throughput, more unbiased investigation of what are the factors that regulate the function of these cells. So to just dive into that a little bit more deeply, we know that there are cell populations within the brain, in particular within the hypothalamus, that are absolutely essential for the regulation of body weight. And we know that these cells respond to certain hormones and nutrients and other metabolic factors, and also that they're connected with other cell populations. And in response to these inputs, the cells produce and secrete neuropeptides that, depending on the cell population, can regulate food intake. And the main strengths of this model system is that it's scalable. So we can produce millions of these cell populations that are actually quite rare in both mice and in humans. If we derive these from human pluripotent stem cells, they are on a human genetic background, and we can control that human genetics using targeted editing tools such as CRISPR, TALENs, or zinc fingers. And we also have environmental control of these cells. So we can isolate them, we can mix them together in known combinations, and we can expose them to desired hormones or other factors at known time points and concentrations. So kind of to harness the strengths of these systems, there's a few lines of investigations within our group that I just thought I would put up here to demonstrate the versatility of what one can do with it. So you can use the scalability and environmental control of this system to screen for new anti-obesity drugs. You can really dive more deeply into the molecular mechanisms of metabolic signaling. So how do these cells respond to individual signals or combinations of signals? Um, we can look at the cellular and molecular consequences of manipulating genes that are associated with obesity to see what are the consequences in particular cell populations. And then finally, we're also interested in how obesity might tie into neurodegenerative disease. We're doing that in a combination of animal models, but also human stem cell derived in vitro models. So that's just a flavor of what we're doing within the laboratory. But to underpin all of this work, we've turned to human pluripotent stem cells. So many of you probably know about these, but just to ensure that we're all on the same page, these can be derived from two different sources. From the human embryo, so the pre-implantation inner cell mass of the blastocyst, or they can be derived from somatic cells and reprogrammed from either skin, blood, or other tissues to generate induced pluripotent stem cells. And these have the properties of being able to be propagated indefinitely in a pluripotent state, meaning that they can give rise to virtually any cell type within the body, and they can be expanded, so we can have access to large numbers of these cell populations. The key is really getting it to generate the cell population that you want. So if we look at where the hypothalamus is developmentally, this is a sagittal view of the developing human brain. And we know that the hypothalamus is both an anterior neural structure and a ventral neural structure. So based on what we know about the developmental biology and the generation of these cell types developmentally, we can come up with a rational directed differentiation program. You can ignore all of the details, but the important thing is that we can guide the differentiation of these cells down a particular pathway to pattern them into hypothalamic progenitors that go on to give rise to hypothalamic neurons. And so I developed a protocol like this when I was a postdoc, and another group simultaneously developed one, and now there's multiple groups around the world using this system to gain access to hypothalamic neurons. And if we immunostain for the resulting cell populations, we see neuropeptides that are either exclusively produced in the hypothalamus or are highly enriched there. This includes alpha MSH or pro-opiomelanocortin or POMC, and AGRP. So what's important about these cell types is that when we look at the genetic basis of obesity and we look at where are the obesity-associated genes more highly expressed, they're clearly expressed in the nervous system, and in particular within the hypothalamus, which we know is key for the long-term regulation of energy balance. And so we've decided to focus initially on these pro-opiomelanocortin or POMC neurons because of the essential role that they play in regulating food intake. So in particular, these POMC neurons are connected to cell populations that inhibit food intake, and they do so by processing POMC into bioactive neuropeptides, these melanocyte-stimulating hormone peptides, which bind to the downstream melanocortin-4 receptor. Loss of function of either POMC or this receptor leads to obesity in both mice and humans. And furthermore, we know that these cells respond to the adipocyte-derived hormone leptin, which famously also, in the loss of function of this, also famously causes obesity. And humans who are leptin deficient can be effectively rescued by leptin replacement therapy. So we know that the circuit is really important, and our hypothesis is that other either genetic or environmental mechanisms contribute to obesity by acting within the circuit. And so we focused on these cell types for the time being because we know that they're biologically relevant, as we just described. Within our culture system, so these are human stem cell derived neurons, they're robustly produced, so very reliably from different cell populations and also at reasonable percentages. And by performing single cell RNA sequencing, we know that these cells are clearly neuronal, relatively mature, and are transcriptionally distinct from other neuron populations that are derived in vitro. So I just wanted to pause here and give an interim summary of what I've told you about so far. So first, I hope I've convinced you that these human pluripotent stem cell or HPSC-based approaches have certain advantages that can be harnessed in order to better understand obesity disease mechanisms, which in turn could provide the foundation for therapeutic development. The second is that recapitulating these developmental processes in vitro can be used to pattern these initially naive cells into physiologically important cell populations that we can use to gain insight into disease mechanisms, that the melanocortin system is a critical regulator of body weight. And then finally, just something that I thought would be useful for you to consider as I move on to the second half of this presentation, is that given that we have this resource, how would you use it? So what do you think could be the most interesting application for these cells, and how would you go about doing it? So in the next half of the talk, I'll tell you about some of the work that we've done to characterize these cells. So first, if you just take a high magnification confocal look at these cells and you stain for this POMC-derived peptide MSH, you see that it's not uniformly distributed throughout the cell, but that it's present in these kind of discrete punctate it has this punctate-like staining pattern. So this is consistent with the packaging of a neuropeptide into a dense core vesicle for release. So next, we hypothesized that perhaps POMC wasn't just being produced as a full-length protein, but that it was appropriately being packaged into these bioactive peptides. So to gain insight into this, we turned into mass spectrometry. And first, just to give you an overview of the POMC gene, it's famous for being extensively post-translationally modified. So what I'm showing here in blue are all these dibasic cleavage sites, which are areas where prohormone convertases can come and chop up the POMC into smaller peptides. And in green, I've shown you where some of these biologically relevant peptides are. In particular, there's alpha MSH, which is classically thought to be the principal stimulant of the melanocortin-4 receptor. There's also its precursor, which lacks this N-terminal acetyl group and is known as des-acetyl alpha MSH. And then finally, there's beta MSH as well. So beta MSH simply does not exist in rodents, which lack one of these dibasic cleavage sites. But there is some evidence that it does actually exist in humans. And this evidence comes from a number of studies, which are now over 14 years old, that were published simultaneously. And there's two different mutations that were identified, one in a highly conserved tyrosine residue and another in one of the dibasic cleavage sites. And in both of these cases, the loss of beta MSH was associated with a extremely higher BMI in individuals that were affected. So this suggested that beta MSH, even though it doesn't appear to be present in rodents, might actually play an important role in human body weight regulation. So to gain some insight into this and to see if actually our culture systems might make beta MSH, we used liquid chromatometry and tandem mass spectrometry, which allowed us to look at both the cell pellets, so the cell bodies of our human hypothalamic neurons, which might contain the reservoir of both unprocessed and processed POMC, as well as cell types that have been stimulated by looking at the supernatant, where we might expect to see an enrichment for the processed form of these peptides. And what we identified was that indeed in our culture system, we could find that POMC was appropriately processed into all of these appetite regulatory peptides, including beta MSH. And to confirm that this is actually true, we then looked in primary fresh human brain samples, which we're very fortunate to gain access to via the Cambridge Brain Bank, and found that both in the regions that encompass the medial-basal hypothalamus and paraventricular nucleus of the human brain, we found exactly the same sequence of beta MSH and also alpha MSH and the beta endorphin variants that we observed in our in vitro culture system. Whereas in control human brain regions from the cortex, we couldn't observe any of these peptides. So this led us to wonder how abundant are these different POMC-derived peptides actually? So given that they exist in the human brain and in vitro, which might be the most physiologically important? And what we found was actually really surprising. So we had a very difficult time in our culture system actually reliably even detecting alpha MSH, whereas we could detect quite a lot of its precursor, desacetyl alpha MSH, and that was produced in roughly equimolar quantities to beta MSH. So this, of course, could just be a consequence of culturing these cells in vitro. They might be insufficiently mature. So returning to, again, the primary human brain samples, we saw exactly the same pattern. So this really suggests that the principal forms of MSH that are produced from POMC are this desacetyl alpha MSH and beta MSH, and that there's much less alpha MSH that's actually detectable. There are, of course, caveats to this. It could be that alpha MSH is acetylated immediately before release, and so the extracellular concentration that's available to bind to the melanocortin-4 receptor could be a lot higher than these analyses are suggesting, but it's still very provocative, and it changes the way that at least I think about melanocortin signaling in humans. So then finally, we wanted to know, is this MSH production dynamically regulated? So here we can take advantage of the fact that we have access to live neurons and we can compare them pre and post stimulation. So that's exactly what we did. We looked at the production of MSH from POMC under standard conditions, and we looked at the various forms of POMC-derived neuropeptides, normalized these all to 1 under standard culture conditions, and then we expose these to leptin for a long enough period of time for leptin signal transduction to lead to increased POMC transcription, translation, and processing. And what we found was a robust and highly significant increase in the production of all of these neuropeptides in response to leptin. So this is really exciting because it's, to our knowledge at least, the first time that it's been demonstrated that these human POMC neurons are responding to leptin much in the same way that mouse cells are doing. But more importantly, we now have access to a culture system that will allow us to screen for factors that can increase the production of this neuropeptide that we know suppresses food intake. So if we could find a way to do that in a specific and potent manner, we might be able to increase the tone of MSH that's present in the brain and therefore activate these pathways that inhibit food intake, and therefore lead to less food intake and hopefully weight loss. So one system that I'll just tell you about at these are among the last slides that I'm going to show, are some of the tools that we're exploring now to increase the throughput of this system. So in particular, we're using high content imaging for the production of POMC. So to do this, we're using a high content confocal imaging system, the Opera Phoenix, and we're immunostaining cells for POMC, which is shown here in this bottom figure. You also see it behind me, inverted. Also neuronal markers and nuclei. And by combining these, we can segment these images, isolate the POMC immunopositive cells, and quantify the levels of POMC production. And by doing that, we can compare POMC production in the context of control conditions. So here we're showing the distribution of POMC intensity in POMC neurons. And the advantage is that we can do this across thousands of cells in a single well. We can then expose these cells to leptin, and indeed we're able to recapitulate the effects that we saw using targeted mass spectrometry. And we can also extend this now to look at candidate compounds. And we found some candidate compounds that can even more potently stimulate POMC production than leptin can. So we're very excited about the potential of this particular approach. So I know I've been talking for a long time. I just wanted to wrap things up and pose a few questions for discussion. So the first of these is, I've highlighted some of the opportunities, but what are some of the caveats that we need to consider when working with these human pluripotent stem cell derived cells? And there are many, and they're important to keep in mind. The second is, what technologies or approaches could be used to further increase the utility of these cells? there's a number of new technologies that have recently emerged, and how could we either use them or marry them with other technologies to expand the potential for the discovery of new disease mechanisms or treatments. And then finally, what are the different cellular phenotypes that we can observe in vitro? And importantly, how can we link a phenotype that's observed in vitro to an organismic phenotype that we might ultimately care about, such as obesity? So with that, I'd like to really thank you for your attention and acknowledge all of the people who've contributed to this work. I'm really fortunate enough to have a fantastic group of people in the Merkle lab to work with. I particularly want to point out Peter Kirwan and Magda Jura, who have now left the laboratory, but whose work has really been the basis of a lot of what I've talked about today. My former advisors and collaborators who helped with the generation of the hypothalamic differentiation protocol, and also my collaborators at Cambridge and further afield who've helped with the characterization of POMC neurons within the laboratory. So thank you very much. I hope you found this useful, and I'm looking forward to catching up when we have a chance to have a discussion. Thank you.